So hello everyone, welcome to today's webinar. I'll just get my presentation going. Just give that a moment to load. So good afternoon or good morning depending on where you are. My name is Emily Perry. I'm the Ensembl Outreach Project Leader and my job is to in many different ways disseminate information about Ensembl and what we're up to. And one of the things that we do is we deliver webinars like this as part of the EBI training webinar series on Wednesday afternoons. If you're in the webinar interface, you should see a section that says materials. If you would like to get a copy of the slides, you can download them there. We'll also send them, you'll get a follow-up email after this webinar. So if you do want to have a copy of them, and you haven't downloaded them here, you'll get it there as well. And we'll also, I am recording this, so we'll post a copy of the video online later as well. So if there's anything you miss and you want to find it again and go back over it, you should be able to. There's also a little chat box. I've muted everyone's microphones, but if you go into the little chat box while I'm delivering the webinar, you can type questions in there. I've got some colleagues who are with me, so Helen Sparrow and Ben Moore from my outreach team, and also Mirto Costadima, who heads up the regulation team, who produce all this data, are all in the chat box ready to answer questions during the webinar. But when I get to the end of it, if there's any questions that we haven't answered, I can go through some things on the screen as well. The chat box doesn't have any threading, so if you're answering somebody else's question or we're answering your question, we'll try and use your name so you know who we're talking to. Otherwise, it can get very confusing. So what I want to talk about today is epigenetics in gene regulation. I want to talk about some of the laboratory methods we use to study epigenetics, because I think it's really important to understand how we produce the kind of data that we're looking at bioinformatically in order to understand what it actually is. And then we'll talk about Ensembl Regulation. So we'll talk about the data we have, where it comes from, and what we do to produce, to process that and produce predictions of things like promoters, enhancers, insulators, and things in the genome. So epigenetics is the study of heritable genetic changes without any changes in the DNA sequence. And this is known to regulate gene expression, so it's really important. And epigenetic change is known to be related to cell differentiation. So if we look at this hematopoietic cell lineage, you can see that there are many different cell types that are all derived from a single stem cell type. And if we compare two different cells of this lineage, we can see that they carry out very different functions. They are morphologically very different. And these come down to the fact that they both express completely different genes, which is due to the fact that they have different epigenomes. And one of the ways people like to, a very old but I think very illustrative way of looking at this is that the stem cell is like this little cell at the top of this marble run here, and as it moves down the marble run, it gets to decide which route it's going to take. And if it takes one route, it will get fixed being one kind of cell. If it takes a different route, it will get fixed being a different kind of differentiated cell. And the different routes are the different epigenetic options. So the methods, there are lots of biological methods that we use to study gene regulation and different kinds of features that we're looking at in Ensembl Regulation. So we look at histone modifications, which we study through ChIP-seq. We look at transcription factor binding, which we also study through ChIP-seq. And we look at open and closed chromatin, which is studied with DNase sensitivity. So if we imagine a gene, so here we have a gene on our DNA, so the red is a gene, and everyone's quite happy with it. The 5' it's 5', I got that wrong for a moment. At the 5' end of the gene, we have a promoter, which is a region of the genome, which is necessary for the gene to be expressed. And we know that RNA polymerase can bind to that promoter, and then it can move along the gene and lead to the gene being expressed. But of course, it can't do that by itself. It needs help, and it needs help from transcription factors. So all these transcription factors bind to the promoter, and only then is the RNA polymerase able to bind. But it's still not that simple, because there might actually be another region of the genome which is further away, which is the enhancer, and that also needs to bind transcription factors. And it's believed that these two regions of the genome physically are brought together because DNA is this flexible polymer, that the two regions are brought close to each other, transcription factors all work together, and only then can we have this RNA polymerase binding. So transcription factors are absolutely necessary for transcription, and combinations of epigenetic marks can affect the ability and probability of transcription factor binding at these particular sites. Now, transcription factors like to bind to particular sequences, which are known as transcription factor motifs. So these are short sequences that attract transcription factors, and they're scored out of 1 for strength. So a score of 1 is a really strong transcription factor. A score of 0 is a really weak transcription factor binding motif. And we pull in transcription factor motifs from Jasper. And there's a URL that you'll be able to get from the download of the slides here. And you can go and explore transcription factor motifs there. And you can see this is what a motif looks like. We have the probabilities of the different bases at each position. So you can see at the first position, you can have pretty much any base. Whereas at position number 3, if you've got anything other than a cytosine, that's probably not going to attract the transcription factor very well at all. We also are looking at histone modification. So this is a diagram of a histone. A histone is a very large protein, which is an octamer. It's made up of 2 of each of 4 individual monomers. So they are H3, H4, H2A, and H2B. And they're this really big monomer, and the DNA wrap octamer, and the DNA wraps around them twice. And so that sort of packages up the DNA. And a really important point about histone proteins is they have these massive long tails that come out of their N-terminus. And when the DNA is wrapped around them, the tails actually dangle out away from the DNA. And these tails are rich in arginines and lysines. And the thing about arginine and lysine is they can be covalently modified. So they can have methyl groups and acetyl groups added to them. And we find that they are covalently modified with these small molecules, and this can affect transcription. There's a standard nomenclature that's used for describing this. So if we consider this lysine here, so this is lysine 36. So if we start at the N-terminus, it's position 36 on the protein, it's a lysine, and it's on histone 3. So we would describe this as it shows here, H3K36. And then if it has 3 methyl groups added onto it, then we would put Me3 for trimethylation. We would also have Me1 or Me2 for mono or dimethylation, or AC for acetylation. And there's what's known as a histone code. So depending on the modification that occurs on the different amino acids on the N-terminus of these different histones, this can increase or decrease the probability of transcription. And we know that these modifications work in concert, that we'll get a number of different modifications occurring on a single histone, and we'll get clusters of histones which are similarly modified. So the technique that we use to study both transcription factor binding and histone modification is ChIP-seq. So ChIP-seq is a technique for studying DNA binding proteins, which is what we're talking about here. So if we imagine we've got DNA which is bound to this DNA binding protein, such as a histone with a particular modification, or a transcription factor. We then covalently cross-link the DNA binding protein to the DNA, and we shear the genome. We cut it up. There are different methods for doing this. Some people use enzymes, some people use sonication, which is basically blasting it with sound waves so it just breaks. But you end up with these short chunks of DNA, some of which will be bound to your DNA binding protein and covalently bound because of the cross-linking that you've done. The shearing tends to be completely random. It's non-specific shearing, so you can end up, if we look at the diagram I've drawn here, you can see this protein has got sort of approximately an equal amount of DNA bound to it at either side, but if we look at this one over here, you can see that we've got a big long tail coming out of one end and hardly any DNA bound to the other end, and that's one of the consequences of doing random shearing like this. You then use an antibody to your protein of interest. So this could be an antibody to your transcription factor, or it could be an antibody to the modification on your histone. And you pull out the protein that you're interested in, and of course, because it's covalently bound to the DNA, you get the DNA as well. You then remove the cross-links, wash, get rid of all the proteins, so all you're left with is the DNA. And you can then sequence these fragments and compare them to the reference, map them on, and you'll see everywhere in the genome that the transcription factor was bound or that the histone modification occurred. One of the questions people always ask when they're looking at ChIP-seq peaks is about the size of them. So if we look at something like a binding motif for a transcription factor, this is the binding motif for CTCF. It's quite a large binding motif at 19 bases. Some of them are as little as 4 bases. This is about the size of the protein itself, and it would be about where the protein is bound. But when we do the ChIP-seq fragmentation, we actually get much bigger pieces of DNA than that 19 bases. So we get about 200 bases in each fragment. And of course, they may be off-center. They're not necessarily right in the middle of the binding. You won't find the motif in the middle of the fragment. You may find the motif right at the end of the fragment. The read length, when you're doing the sequencing, a good read length is considered to be about 50 bases. So what you actually could end up with, if you consider your little 19 base motif, is sort of 50 base long reads scattered all over a region 400 bases wide, so going 200 bases either side of your actual binding site, which means that the peak you would call through ChIP-seq is often much larger than the binding motif, and can, through chance alone, be slightly off-center as well from where the binding motif is. And of course, we're interested in the actual binding compared to the binding motif, because the actual binding will differ between cell types, whereas the motif is something that is present in every cell with that sequence. So we're also interested in open and closed chromatin. So this is just another way of depicting my histones. This time I've drawn them as these little beads, these yellow beads, and we've got the DNA going twice around each one. And you'll see that over here, I've got lots of histones very close together, whereas over on this side here, the histones are much more spaced out. And it's known that transcription factors and polymerase and things are more likely to bind, and therefore are more likely to get transcription in areas where the histones are much more spaced out. So the regions where we've got the spaced out histones are known as open chromatin, and it's transcriptionally active. The regions where they're all bunched together is closed chromatin, and they are inactive. We study this with DNase hypersensitivity. So DNase is an enzyme that can digest DNA. And if we treat chromatin, so DNA that's bound to histones, with DNase, we find that it can get in and it can cut far more in regions where there is less in the way of histone. So open chromatin gets chopped up a lot more, closed chromatin tends to stay largely intact. This means that we can purify the DNA and we can sequence those regions that were chopped up really small. And again, we compare to the reference and see where we had this open chromatin. So we have these different techniques, we have these different kinds of data that we're looking at, and this is what we're using in Ensembl Regulation. So we're trying to annotate the genome with all these different features that play a role in the transcriptional regulation of genes. So we're looking at open and closed chromatin, we're looking at transcription factor binding, we're looking at epigenetic marks, and we're also looking at where RNA polymerase binds, which we also study with ChIP-seq. Currently, we have data in human from ENCODE, from Roadmap Epigenomics, and from Blueprint. In mouse, we have data from ENCODE, so we don't produce any of this data ourselves. We pull it all in from different external sources. And in the future, we plan to get data. We're part of the FANG consortium, who are looking to do similar kinds of experiments with different agricultural livestock animals. And there's also a zebrafish project underway as well. So if we look at the data sources that we have, so we have data from ENCODE and Roadmap Epigenomics in humans. So we have ChIP-seq, histone modifications, transcription factor binding sites, and RNA polymerase and DNA sensitivity available. And we have this for 19 cultured cell lines for ENCODE and Roadmap Epigenomics. So these were massive international projects where there were lots and lots of these kinds of experiments done on different cultured cell types. They also did some experiments with mouse in ENCODE, and again we have ChIP-seq, histone modifications, transcription factor binding sites, and RNA polymerase, plus DNA sensitivity. We have slightly less data from Blueprint epigenome, so we only have histone modifications and DNase sensitivity, but this was using 26, we have data from 26 primary cell types that are part of the hematopoietic cell lineage. So instead of cultured cell lines that you might use in the lab, these are primary cell types that are taken directly from human tissue. So we have, because it's a lineage, it's quite interesting because you can compare how things would have changed over the cell differentiation from Blueprint. We don't have all of the cell types that each of those projects have created data from, because we need a certain amount of data in order to produce what we call our regulatory build, which I'm going to go over in a moment. So we display cell types that have, at a minimum, CTCF binding. Of course, this isn't available from Blueprint, DNase data, and these three major histone modifications, so H3K4 trimethylation, H3K27 trimethylation, and H3K36 trimethylation. We display all of the transcription factor binding sites, histone modification, everything that's known in these cell types from those sources, and we process these data to predict activity. We also have track hubs, which I will mention briefly at the end of today's presentation, where you can add further data from these different sources. So we take the raw data from the various sources, we process it to predict the positions of regulatory features such as promoters, enhancers, insulators, and we predict the activity of the features in the different cell types. And you can view this in the genome browser. You can also access it through our APIs and through BioMart. So this is kind of the start point we're going from with our data. So we have the kind of read count at any given position. So if this is a genomic region, we have the read count for the ChIP-seq all the DNase sensitivity data across the region, and we call peaks on these reads. So we can see that here, where we've got lots and lots of reads, we've called this as a blue peak here, this particular histone modification. And what we do with this data is we look for patterns. So we would say, well, this looks like a region of interest. Our pipeline would identify this, would identify that there are a number of different features occurring together in one place. And so then it looks around the whole of the genome and it tries to identify regions where we see the same patterns repeated. And it finds numbers of different regions all over the genome, all of which have this similar collection of peaks of histone modifications and transcription factors combined together. And it's able to say, well, we've already got data that suggests that some of these are known promoters. Based on the idea that some of them are known promoters, if we see the same pattern elsewhere in the genome, it can then make the prediction that all of those other regions are probably also promoters as well. Now, it's not so simple as being able to say, well, there's one pattern, and that pattern is always found at all promoters, because there'll be another pattern which also has a known promoter, which we can also say, well, they're promoters too. And there'll be another pattern which is again different, which is again got some known promoters which match it. So there are actually many, many different patterns across the genome that are associated with different kinds of activity. So just because one promoter has certain patterns and certain transcription factors, there'll be another promoter which is equally valid, which has completely different patterns found on it. So this produces what we call segmentation, which is the next step in our process, and this is done for a single cell type at a time. We then take the segmentation from the single cell types and we combine, oh sorry, I missed a step. Just to point out that this is completely blind. Our algorithm doesn't actually know what the patterns it is looking at are. So it doesn't know whether a histone modification is activating or repressing, for example. All it does is it says, I can see this pattern, I can see that pattern, let's match them up. Later analysis, when we go through this afterwards, we find that we are getting activating modifications at activating segments and repressing modifications at repressing segments. So it's proving that this algorithm, even though it's doing it by machine learning, even though we're not telling it what to think, it is actually thinking very sensible things. We believe that it is working properly. So we take the segmentation from the different cell types and we line them up next to each other. And we can see that we'll get some matches between cell types, but we'll get some mismatches because, of course, each of these cell types are doing something ever so slightly different. So what we do is we then take a binning system and we go through, and every 200 bases we'll make a bin and we'll say, what is in this 200 bases? Is there anything in any of the cell types, is there any evidence that there is some kind of activity occurring? And we can say, well, in this 200 base bin that I've identified here, we have some promoter activity. And then we move into the next region and we ask the same question. And we keep moving across and across and across in these 200 base bins, and some regions we will identify as having a particular kind of activity, and other regions we will identify and we'll say, well, there's nothing really happening there. And this creates what we call the regulatory features. This is where we're saying, this is approximately on the genome where we believe that there is a promoter, an enhancer, an insulator. And these are genomic features which are independent of activity. We're just saying that's what that part of the genome does. Their activity is then predicted in the different cell types. So we'll say, okay, we have a promoter here. In cell type 1, we believe it's active, so we'll colour it in red. In cell type 2, we don't think it's active. So we'll colour it in grey with the sort of hashed red on it so we know that it's still referring to a promoter. Inactive in 3, active in 4. And this is what you'll see across the different cell types. Through this analysis, we've covered about 300 megabases of the genome, identified about that amount in carrying out these different kinds of functions. So about 10% of the human genome we've identified as being involved in regulatory activity. Just to point out a couple of things, a couple of provisos. Currently, we don't link any of our regulatory features to genes. We show you where they are, and we let you make your own inferences. Obviously, linking, if you find a promoter at the 5' end of a gene, that's a fairly logical inference to make. But things like enhancers, which are a bit further away, which would probably need experimental analysis to determine which genes they belong to, we're not able to identify that with the way that we build these features. We also don't link to gene expression. There's cell line specific regulation data, and you can get tissue specific expression data, and again we let you make that inference yourself. It's very complex data, and it is still in its relative infancy. There's no kind of comprehensive database which identifies where the promoter of every gene is or where enhancers of every gene are. But what we're trying to do is provide a starting point, a kind of booster point, so that people can do those experiments. People then say, well, are they real? Is this promoter real? Well, all our predictions are based on real biological data. They're not just sequence analysis. They are based on these kinds of experiments, the ChIP-seq and the DNase sensitivity experiments. And we have strong evidence to suggest that they're doing what we think, but they've not been experimentally validated. Nobody's cloned them alongside a gene and done the wet test that would determine. And of course, with every cell type we get, with every extra experiment we get that we can add into our pipeline, the better it will get at doing these predictions. The number of cell types we have, while it is vast, is nowhere near the number of cell types actually found within an individual. You can also get, if you're familiar with BioMart, you can also get regulatory data through BioMart. So you can look at motifs, regulatory features, and evidence. You can filter by locations, by cell types. And you can get things like locations, regulatory feature IDs, activities, stuff like that. We can also give you consequences of regulatory features, of variants on regulatory features. So we will tell you if variants fall within regulatory features. We'll tell you if it falls within a transcription factor binding motif. And if it changes the strength score of that motif, we will give you that as well. And this is shown for all known variants. But also if you run the Ensembl vet on variants, you can get those predictions as well. So we're going to have a look at a region of a gene called LIMD2. We're going to find regulatory features and explore what cell types they're active in, and explore what evidence there is to show this. So I am going to hop out of my presentation. I will quickly just open the webinar interface. I can't see the chat box or anything while I'm presenting, so it's very hard to see it. So I'll just see there's nothing. Looks like Meto's been answering all the questions. So it doesn't look like I have anything particular to help with. So I will just carry on and start having an explore. So just hide that again. So here I am on the Ensembl homepage. If you haven't been to it before, we're at ensembl.org, and there's lots of useful features on the Ensembl homepage. Just to point out a few useful things, there's a link here to help and documentation. I do recommend going and having a look there if you would like to find out more about some of the background behind data. You can explore particular genomes, but I'm just going to go straight into the search box here, and I'm going to search for the name of my gene, which is LIMD2, and hit return, which takes me to the search results. The search results have faceted options, so it will show me the human gene first, because the search knows that the most popular thing that people ever search for are human genes, but you can use the facets at the side to narrow down your search. And instead of clicking on the identifier for the gene, I'm going to click on this link just below it here, which is the location. So here we are in the location view, and you can see we've got the gene LIMD2 and its various transcripts. What I'm going to do is I'm just going to use this box here to drag out a little box and jump to region that's ever so slightly bigger than the region we're currently looking at. Whenever we're looking at regulatory features, we probably want to look a little bit outside of the genes, because they're not always, they can sometimes be within genes, but they're usually outside of the genes. So now we have our LIMD2 gene in the middle, and we've got some other neighbouring genes alongside. LIMD2 is a negatively stranded gene. I know this because I've got these little arrows giving me the direction of transcription, and it's also depicted below this blue bar. And so all negatively stranded genes will be depicted there. So if I'm looking for a promoter of this gene, I want to look over this side. So if I scroll down, you see I've actually got this massive promoter which spans pretty much the whole of the gene. Sorry, I know that it's a promoter. I do this all the time. I'm looking at this big red thing and saying it's a promoter. I know this because I work with it, so I see these colours all the time. But there is a legend at the bottom here, which tells me that red means promoter. This sort of pink is the promoter flank, so I've got flank on either side. I've also got a couple of enhancers over here and some CTCF binding sites shown in turquoise here. I've got these black lines within the promoter, and these black lines indicate the transcription factor binding motifs. So if I click on this promoter, you'll see I get a little pop-up. It gives me a stable identifier of the promoter, which is ENSR, so it's Ensembl R for regulation, and then a long number which indicates it is unique to this particular promoter. I've got its coordinates. It tells me it's a promoter just in case I haven't looked at the legend, and it also tells me what all of these transcription factor binding motifs are, and what their scores are. And I could click, I will click in fact, and it'll open in a new tab for me. This is a really short transcription factor binding motif. It's just 5 bases long, and it takes me to Jasper, and so I can see what those bases are. I will get rid of that and go back to my menu here, and I'm going to click on the stable identifier. And this takes me to what we call the regulation tab. So Ensembl has a system of tabbing where you get these little tabs appear along the top, which can mean that you can kind of switch back and forth between different features of interest very easily. So I could go back to my location, go back to my regulatory feature quite simply using these. The regulation tab contains all of the bits of information about the that you can possibly find about the regulatory feature that we have available. So I've got a little summary up here. It says active in 22 out of the 38 cell lines that we have available. And if I look at the diagram below, you'll see that I have activity. So this is the MultiCell. This is the sort of where we think the feature is. But then in some cell types you can see it's listed as inactive. It's this grey hashed box. And in others it's active. It's shown in red. We've even got this dark grey, which you'll see means it's repressed. So it's not even just an inactive promoter, it's a promoter that's being actively stopped from transcribing. And we also have a purple one, which is poised, which means it's got some of the things that it needs to be active, but not all of the things, and it's ready for the extra stuff to come along and get it ready to start transcribing, but it's not going to do that yet, not until it's got more stuff. So you can see all the different cell types. If I want a bit more information about what's going on and what the different features involved are, you can see that when I clicked on these, it showed me the attributes, so the different transcription factors and histone modifications that were occurring at that location. If I would like to see it in more detail, in that cell type that is, if I want to see it in more detail, I will go to Details by Cell Type, this button at the top here. Now this page only shows me one cell type by default, which is this HuVec cell line where, from ENCODE, where we've got an active promoter. But I can show more cells if I want. If I click on this Select Cells, it allows me to pick more cell types. And I'm going to pick HeLa S3. The reason I'm picking HeLa S3 is because I have remembered from the previous page that it was inactive in HeLa S3, and I would like to compare an active to an inactive cell type. I could also turn them all on or all off if I wanted, but I'm just going to turn on HeLa S3. I click on the tick, it will now load. So you can see I've got my HeLas where it's inactive. I also want to see all the possible evidence available, so I have this second button, Select Evidence, and this time I am going to say all on. Everything that's available, any transcription factor, any histone modification you've got, I want to see it because I want to see as much evidence for this as I can. So if I save and close that one, it's going to load, and we'll see that these two cell types are very different in terms of what they've got bound to this particular promoter region. So in our HeLa cells, you can see in both cell types we've got CTCF binding, but that also corresponds to these active CTCF binding sites. So you can see the legend over here. So this green is CTCF binding. The sort of little arrows top and bottom are the kind of peak of the peak. So you can see we've got two of these in the HuVecs and two of these in the HeLas, which correspond to these transcription factor CTCF binding predicted regions, and also DNase sensitivity. But we've also got further DNase sensitivity across the HuVec cells. We've got a transcription factor bound, which is max. We have some histone modifications, so we have some activating H3K4 monomethylation and trimethylation in this region. There's H3K36 trimethylation a bit further out. And the real kicker, the real convincer that this is probably an active promoter is this DNA polymerase 2, RNA polymerase 2 even, binding that we've got across most of this promoter. If we compare that to the HeLas, we haven't got anywhere near the same level of activating histone modifications, polymerase binding. So we can see why this was probably predicted as active in the HuVecs, but inactive in the HeLas. I'm just going to hop back to the Location tab, because if you're interested in looking at this over this kind of thing over a wide region, rather than just looking at a single regulatory feature, you can do that in the Location tab. So if we go into the Location tab, as we saw before, we've got our promoter, but we've also got some more features a bit further out as well. And if I go into this blue button here, which is Configure this page, you'll see that I now have a menu with loads and loads of options of tracks, which are basically data types that we can plot against the genome, that I can display against my region of interest. And I'm going to go into this regulation section. I'm going to go into regulatory features first of all. And I'm going to turn on my HUVEX and my healers again. If I just click on the box, it switches that view on for me. I'm then going to go into Open Chromatin and Transcription Factor Finding Sites, which is underneath it here. And you'll see I now get my cell lines listed along the top, and I get my transcription factors listed down the side in a kind of matrix. And where they meet, I can click on that, and that will turn that particular track on in that particular cell type. I also have a select all for a transcription factor I could click on, and I have a select all for my cell types. I'm going to go select all for my 2 cell types again. Where'd it go? And you'll see it's chosen a track style. So when you turn on tracks in Ensembl, they can appear in different styles. Because I've switched these on, it's chosen for me that I want it as peaks. So it'll just show me the top of where it thinks it's bound. If I wanted to see the kind of number of reads, then I could put it on signal. If I wanted to see that as well as the peaks, I could put it on both. But I'm going to leave it on peaks just because it gets very busy. I'm also going to go into histones and polymerases, where we'll see a matrix that's very similar. Because we also have histone and polymerase data for the blueprint cell lines, you can see we've got a lot more data, a lot more cell lines along the top here. And so I'm going to turn on my all for my healers and my HUVEX again. And now if I click on the tick in the top right hand corner, it's going to load this for me. It's going to load a lot of data for me. But you can see all across the whole of the region in a very similar display to how I saw it before. I've got the MultiCell followed by the HUVEX, and I've got CTCF, DNase 1 sensitivity, max, just as I saw before, and I've got my histone modifications and my polymerases. So I can see where all of these peaks occur, not just in reference to the promoter I was looking at earlier, but also further away in the genome as well. So I can see what corresponds to this enhancer over here, which is inactive in both cell types. So that's kind of looking at the regulatory build as we have it. I'm going to just hop back to the home page because I would like to show you track hubs. So there's a link here at the bottom of the page which says Ensembl supports data from external projects through track hubs. And if you're interested in looking at things like all of the data from Blueprint or from ENCODE or something like that, you can do so using the public track hubs. So this will take me, gives me a bit of information, but I'm going to hop straight to this link which says Track Hub Registry Search for Human. And I'm going to do a simple text search for Blueprint. I'm going to do search, and it gives me 2 options of hubs. I'm going to pick the second one and say attach this hub. So this is data that is stored externally but can be shown in Ensembl. If I now hit, so it says your hub was attached successfully. If I hit configure your hub, I now get back into a region view. It's a different region to where I was looking at before, but you can see it's added Blueprint hub to my menu on the left. And it's also telling me that there were 2,254 tracks available as part of this hub. So you can see we've actually got many, many, many hubs available in the track hub registry. If each of those have many thousands of tracks available, you can see how that would really slow everything down and make it very hard to find the things that you're interested in, which is why we have these as separate things that you can add. If I go into Blueprint region, you'll see that I have the kind of matrix display that I had before. So I've got kind of different cells, and I've got different histone modifications, which I can treat just as I did before. The big thing that's different is you can see in the boxes there are these numbers. So in the top corner we've got a 0, and in the bottom corner we've got a 14 in this particular one. If I click on it, you'll see that I get 14 tracks available. So these will be individual experiments that were done. So the groups like Blueprint, who we got this data from, they didn't just do one H3K27 trimethylation ChIP-seq. They did multiple replicates of it. And so now I have the option of, instead of just adding a kind of summary data, which is what we had in the main thing, I can instead add the individual experiments, and I can do them one at a time. And you'll see when I do that, it then says 1 out of 14. So the top corner is how many I have selected. The bottom corner is how many are available. And you can explore the different track hubs from these different sources, and you'll see it's now loading. It's taking its time because I've now added quite a lot of data to the browser. It does slow it down a lot, which is why we only recommend adding these if you know you definitely want to use them. But it's added all this extra data. So, see if there are any questions that still need to be dealt with, but it looks like everything's fine, which is good. And now I'll hop back into the presentation and just sort of wrap up everything that we've done today. So, if you thought that this was a useful webinar, if you are interested in having Ensembl courses, we can come out to you and teach an Ensembl course at your institute for free. Aside from the trainer's expenses, we can do browser courses, and we can also do API courses. If you send us an email to helpdesk@ensembl.org, we can discuss your requirements and how we would do this with you. There is absolutely tons of help and documentation available. Many of you will have found this course through EBI Train Online, which is a fantastic resource with loads and loads of different kinds of training on bioinformatics in general, as well as Ensembl. We also have some tutorials. We have this, the video of this webinar, we'll put this up on YouTube, which has a number of other short tutorial videos available. And there's also a YouTube channel for anybody who happens to be in China. And you can always email us to ask for help with anything Ensembl related. You can email us privately to helpdesk@ensembl.org, or you can email the dev mailing list. If you join that, then that's a public mailing list, and that's more aimed at people who are working with Ensembl data in a programmatic fashion. You can follow us. Again, some of you may have found out about this webinar through our social media. We have Facebook, Twitter. We also have a blog. And we have publications. So we have a main publication that comes out every year. And we also have, there's some publications on the regulatory build. So there's a paper from genome biology, which explains how the build was produced. And there's a paper in database, which tells you a bit about how the data can be accessed and some of the interfaces that were produced. This was our team at our retreat. This was over a year ago now. Hopefully we'll get a new picture like this soon. And just to say thank you to everybody who's involved in Ensembl, and also our funders who allow the project to take place. If you're interested in more webinars, the EBI do have a training page where they list different webinars that are available. We're giving another webinar in a few weeks because we're going to have a new release out soon. So we'll talk a bit about what's involved in that new release. And there's a webinar from the ontology lookup service. I don't know much about it, but if you go to the website and have a look, you can find out a bit more. And all future webinars will be announced on that page. So I will quickly hop back out and see if there are any more questions that want to be answered, or you can ask questions now. There is an option to do a little hands up thing, and I can put your microphone on, but in my experience, sometimes that just gets a bit, it doesn't work very well, and you can't hear people very well. But we can try. Okay, if there's no questions, then thank you very much for attending. If you come up with any questions later, please do send us an email to helpdesk@ensemble.org. I hope this was useful to you, and enjoy the rest of your day.